In this lecture, we're going to just discuss several hereditary diseases as well as some molecular methods to diagnose these diseases. At the end of the lecture, you should be able to describe Mendelian patterns of inheritance as exhibited by pedigree diagrams, discuss the inherited diseases, including causes and symptoms that we go over in this lecture. Give examples of and interpret laboratory methods designed to detect single gene disorders, discuss non-Mendelian inheritance patterns, and give examples of these types of inheritance, such as mitochondrial disorders and trinucleotide repeat expansion diseases. Show how genomic imprinting can affect disease phenotype, and list the genetic locations and nomenclature for the discussed diseases. So our models of disease etiology, we have genetic diseases, which are inherited diseases, which we are going to discuss in this lecture. We have environmental diseases, which are usually based on somatic mutations. We also have multifactorial diseases. These are polygenic, meaning numerous genes are involved, plus there are environmental factors that commonly are involved. So we discussed some genetic diseases, and we went over pedigrees and inheritance patterns earlier in the semester. So just as a review, the family history of a phenotype is illustrated using a pedigree. You have your squares, which are males, circles are females, filled in are affected. So just to remember how to read your pedigrees. Here's an example of an autosomal dominant pedigree, an autosomal recessive pedigree, and a sex-linked, and in this instance, an X-linked recessive disorder. So autosomal recessive, autosomal dominant, or sex-linked patterns are observed in single gene disorders, and these are diseases that are caused by one genetic mutation. Prediction of transmission pattern assumes Mendelian inheritance of the mutant allele, but remember, not all diseases are inherited in a Mendelian fashion. Some of the patterns of transmission, we have loss of function mutations, which usually display a recessive phenotype, and the phenotype of these mutations depends on the type of protein that's affected. We also have gain of function mutations, which are less common than the loss of function mutations. In gain of function mutations, new properties of the mutant allele are responsible for the disease phenotype. Mutations include gene expression and stability defects that generate gene products or proteins at inappropriate sites or times. Usually these display a dominant phenotype. We also have dominant negative patterns, which are observed with loss of function mutations in multimeric proteins. And here's a schematic of a dominant negative pattern, disease pattern. So you would have a homozygous individual, which would in this case have a normal phenotype. You also have a heterozygous where you have a mutant allele, and this is going to cause an abnormal phenotype. And autosomal recessive disorders, these are the most frequently observed transmission patterns. The mutant phenotype is not observed in a heterozygous individual. The mutation must be homozygous in order to show the abnormal phenotype. We also have sex-linked disorders, which are more commonly X-linked because relatively few genes are carried on the Y chromosome. X-linked mutations are usually recessive, and males are hemizygous for X-linked genes, so they're more likely to develop the phenotype in an X-linked disorder. Loss of heterozygosity. In autosomal recessive mutations, these can result in an abnormal phenotype in a hemizygous state. Loss of the normal allele, revealing a mutant allele, is called loss of heterozygosity, and we talked about this in the previous lecture on cancer detection. Loss of heterozygosity results from somatic mutations or deletions of the normal allele. So these are not inherited mutations. Genetic testing can be very complex because diseases and inheritance patterns are complex. So you can have variable expressivity, where a single genetic mutation results in a range of phenotypes as opposed to one single phenotype. You have genetic heterogeneity, where the same phenotype results from mutations in different genes. So this includes diseases with multiple genetic components. We also have penetrance, where the presence of a mutation may occur without the predicted phenotype. The study of genetic diseases is a large scientific discipline, and the theoretical underpinning is based on population genetics. Currently, around 4,000 genetic disorders are known, and more are constantly being discovered. Most disorders are very rare and affect one person in every several thousand individuals or even million individuals. Mutations are heritable changes in DNA nucleotide sequences. Mutation in germ cells result in inherited diseases. Mutations in the somatic cells result in cancer and some congenital malformations. Congenital diseases are diseases that are present at birth. They are not always inherited diseases, and an example of a congenital disease that is not inherited is spina bifida. The molecular basis of single gene disorders. Well, usually these are going to cause disorders due to mutations in certain regions of the genome, and these are usually going to be in structural proteins, and these are going to cause diseases such as sickle cell anemia, Marfan syndrome, and muscular dystrophy. Mutations in cell surface receptor proteins, a disease with this type of mutation is familial hypercholesterolemia. Mutations in growth regulators, neurofibromatosis, and mutations in enzymes such as phenylketonuria and severe combined immunodeficiency. Examples of single gene disorders. There are lysosomal storage diseases, factor V Leiden, hemachromatosis, cystic fibrosis, and cytochrome P450. Lysosomes are cellular organelles that degrade cellular products of ingestion through acid hydrolase enzymes. So lysosomal storage disorders result from incompletely digested macromolecules due to a loss of enzyme function. These disorders are usually screened by gene product testing, and the mutations are detected through direct sequencing. Here's an example of several storage diseases. So on the left you have your substrate that's accumulated, and on the right is the disease. So when you accumulate sphingolipids, this is Tay-Sachs disease. When you accumulate sulfatides, it causes Niemann-Pick disease. So depending on what the substrate is, you get various diseases. Genetic lesions in single gene disorders are detected through various hybridization and amplification methods as well as nucleotide sequencing. So these are going to be the more common methods to detect single gene disorders. The first single gene disorder we're going to discuss is factor V Leiden, which was discovered in 1994. The Leiden mutation occurs in coagulation factor 5 gene, which is on chromosome 1, and the mutation A to G at location 1691 is the common mutation that is found. So this causes hypercoagulation, which leads to deep venous thrombosis, and it's treated with anticoagulants. So this genotype is present in the heterozygous form in 4 to 7% of the general population and is homozygous in 0.06 to 0.25% of the population. Prothrombin is the precursor to thrombin in the coagulation cascade and is required for the conversion of fibrinogen to fibrin. A mutation in the 3' untranslated region of the gene that codes prothrombin or coagulation factor 2 located on chromosome 11 results in an autosomal dominant increased risk of thrombosis. Laboratory tests can be used to detect the F2 and F5 mutations. Both of these mutations may be present in the same individual, and if an individual has both mutation, it increases the risk for thrombosis. So this slide is showing a PCR RFLP test to detect the factor 5 Leiden mutation. So in this PCR RFLP, you are going to use a forward and reverse primer to amplify the gene in exon 10. And once you get your product, your PCR product, you're then going to digest with the restriction enzyme MNL1. And in a normal individual, there are 2 MNL1 restriction sites in this region. Now, in the G to A mutation, it's going to destroy one of those MNL1 sites. So you're going to lose that cut site if you have a mutation. So then you can compare the number and sizes of bands on a gel of your PCR product that's been digested with MNL1, and you can see the differences between a homozygous normal individual that doesn't have the mutation, a heterozygous individual, and a person that is homozygous for the mutation. In this example, we have a mutation detection for factor 5 Leiden using the SSP PCR methodology. So again, you're looking at the gene on exon 10, and you have sequence-specific PCR primers. So you have one primer that has a longer ends, which has your mutation, and it's going to generate a larger amplicon. So if you have the mutation, you're going to get a 148 base pair amplicon. If you do not have the mutation, you're going to end up with a 123 base pair amplicon. You can also use the invader system to detect the factor 5 Leiden mutation, where you have a mutant probe and you have your wild type probe. You have your mutation that's present, which is going to generate cleavage, or you have in a normal sample, you're not going to get cleavage. When you have cleavage, you're going to get complex formation, you're going to get your fluorophore and quencher that come into close proximity with one another, and you're going to get fluorescence, which is going to indicate the presence of mutation. Another single gene disorder is hemachromatosis. This is an autosomal recessive condition that causes overabsorption of iron. Iron accumulation causes pancreas, liver, and skin damage, heart disease, and diabetes. Classically, hemachromatosis is diagnosed by measuring blood iron levels, and this disease is treated with therapeutic phlebotomy. So in hemachromatosis, you have a dysfunction of the hemachromatosis type 1 gene, or HFE, or sometimes HLA-H, which is located on chromosome 6. The HFE gene codes for a membrane-bound protein that binds with beta-2 microglobulin and transferrin on the membrane of cells in the small intestine and placenta. So this protein directs iron absorption based on cellular iron loads. In the absence of the HFE gene function, intestinal cells don't sense iron stores and continue to absorb iron into an overloaded state. So most frequently, the observed mutation is a C to Y change at amino acid position 282. This mutation is present in 10% of the white population and is detected using PCR RFLP methodologies. Other mutations also occur in the HFE protein. We have an H to D mutation at position 63 and an S to C at position 65. So here is a schematic showing the HFE protein and the locations of the most commonly observed mutations. Here is a schematic of the PCR RFLP method to detect the C to Y HFE protein mutation at amino acid 282. So in this method, once again, you have your PCR primers, which are going to amplify a region that includes the gene on exon 4. When you have a mutation, in this example, it's going to create an RSA1 restriction endonuclease site. So if you amplify this region through PCR and then cut with the RSA1 enzyme and then run it on a gel, you're actually going to get an extra small band if you have the mutation, because it's going to generate another restriction site. Another single gene disorder is cystic fibrosis, which is an autosomal recessive disease that causes severe lung damage. This disorder affects the cells that produce mucus, sweat, saliva, and digestive juices and causes thick, sticky secretions. It affects approximately 30,000 people in the United States, and respiratory failure is the most dangerous consequence. There's currently no cure, but early diagnosis does improve the treatment strategies. Cystic fibrosis is caused by a loss of function of the cystic fibrosis transmembrane conductance regulator gene, or the CFTR gene, located on the long arm of chromosome 7. This gene codes for a chloride channel membrane protein, and the most frequently observed mutation is a 3 base pair deletion that removes a phenylalanine from the position 508. More than 1,300 mutations have been reported, and there are various molecular tests to identify these mutations, and these include RFLP, PCR RFLP, SSCP, SSP-PCR, direct sequencing, and array technologies. Cytochrome P450 is another single gene disorder. It comprises a group of enzymes which are monooxygenases that are located to the endoplasmic reticulum. So enzymes influence steroid, amino acid, and drug metabolism, these cytochrome P450 monooxygenase enzymes. Over 30 reported cytochrome P450 enzyme variations occur. Enzymes are classified according to the families and subfamilies. So CYP2A6 is cytochrome P450 subfamily 2A polypeptide 6. CYP1A2 and the enzymes in CYP2 and CYP3 families are considered to be the most important in drug metabolism. So the genes coding for these enzymes are located throughout the genome. Genetic polymorphisms of cytochrome P450 genes are unequally distributed geographically and in different ethnic populations. Testing for these polymorphisms is used to predict the response to drugs sensitive to metabolism by this enzyme system. Testing is performed using allele-specific PCR for particular polymorphisms. Multiple P450 genetic variants may be screened using microarray or bead array technologies. So now we're going to discuss single gene disorders with non-classical inheritance patterns. So non-Mendelian transmission patterns. Single gene disorders are disorders with multiple genetic components with non-classical patterns of transmission include gonadal mosaicism, which is a somatic mutation in germline cells, specifically within the gonads, genomic imprinting, which is nucleotide or histone modifications that do not change the DNA sequence, nucleotide repeat expansion, which increases the allele sizes that disrupt the gene function, and mitochondrial inheritance, where you have maternal inheritance of mitochondrial genes. Here are pedigrees showing non-Mendelian transmission patterns. So we have gonadal mosaicism, nucleotide repeat expansion, and mitochondrial inheritance. Mutations in mitochondrial genes. Mitochondria are cellular organelles responsible for energy production. They contain their own circular DNA genome. They're approximately 16,500 base pairs in length and contain 37 genes. Mutations in mitochondrial genes affect energy production, and they're usually manifested as diseases in muscle and nervous system. Human disorders due to mitochondrial mutations include Cairns-Sayre syndrome, Leber hereditary optic neuropathy, myoclonic epilepsy with ragged red fibers, deafness. There's numerous mitochondrial disorders. Here is a schematic showing the mitochondrial circular DNA and some of the mutations within the mitochondrial DNA that are associated with diseases. So in your mitochondrial mutations, you could have homoplasmy, where all mitochondria in a cell are the same, or heteroplasmy, where some mitochondria are normal and others have mutations. And the severity of the disease phenotype depends on the amount of mutant and normal mitochondria that are present. So in this slide, we're showing the detection of the NARP disease that's caused by a mitochondrial point mutation, and this we're detecting through PCR RFLP. So if you have the point mutation, a T to either a C or a G at position 8993 in the ATPase6 mitochondrial gene, the mutation is going to create an MSP1 restriction enzyme site. So you're going to get an extra band once you do your PCR and cut with MSP1 if you have the mutation present. Detecting the disorder KSS or Cairns syndrome, which is caused by a mitochondrial deletion mutation, and this is done through southern blot. So in southern blotting, you take your DNA, you are going to digest it, and in this case using the restriction enzyme PVU2, which is going to cut only once in the circular mitochondrial DNA. But if you have a mutation, you are going to get an heteroplasmy, where you're going to get some mitochondria that have the mutation, so you're going to get that extra cut site, and some mitochondria that don't have the mutation. So you're going to get the two bands, the one large normal band and the smaller band where you have your mutation. Are trinucleotide repeat expansion disorders. These are triple repeats, 3 base pair repeating units, and are referred to as STRs, or short tandem repeats. So during DNA replication and meiosis, short tandem repeats can expand or contract in length. Mutations may occur in coding and non-coding sequences, and 2 well-known examples of trinucleotide repeat expansion disorders are Fragile X syndrome and Huntington's disease. In Fragile X syndrome, you have a CGG expansion in the non-coding region 5' to the Fragile X mental retardation gene, or FMR1. Full Fragile X syndrome has more than 2,000 CGG repeats. Expansion results in methylation and transcriptional shutdown of the FMR1 gene. So symptoms include learning disorders, a long face, large ears, large genitalia, and symptoms usually increase with each generation. So here's an example of the FMR1 gene with your nucleotide repeats. So in a normal individual in the FMR1 gene, you have between 5 and 55 CGG repeats. In a person that's carrying the disorder in this gene, you would have between 56 and 200 CGG repeats. However, in an affected individual, you would have between 200 and over 2,000, up to over 2,000 CGG repeats in the FMR1 gene. So you can do either PCR or southern blotting to detect the CGG expansion mutations. So PCR is used to detect pre-mutations because there's a smaller number of repeats. However, to detect full mutation, that over 2,000 repeats, you need a southern blot because that's going to be a very large fragment. So in PCR, you could see that if you get larger products, it's going to indicate that in that gene, you have numerous repeats. So when you amplify that region, the more repeats you have, the higher the amplicon, the larger the amplicon you're going to get. So when you get a banding amplicon that is indicative of 50 to 90 repeats, that would be considered a pre-mutation. If you do your southern blot, you can actually see the very top band, which would be a full mutation. Huntington's disease is another trinucleotide repeat expansion disorder. It used to be called Huntington's chorea, and this is a rare inherited neurological disorder affecting up to 8 people per 100,000 individuals. It affects 1 out of 20,000 people of Western European descent, 1 out of 1 million people of Asian and African descent, and it takes its name from the Ohio physician George Huntington, who described it in 1872 in his first medical article. The prevalence is 5 to 8 per 100,000, and it varies geographically. So Huntington's disease is caused by a trinucleotide repeat expansion in the Huntington gene located on the short arm of chromosome 4. The sequence CAG expands from 9 to 37 repeats in a normal individual to between 38 and 86 repeats in a diseased individual. And this is one of several polyglutamine or polyQ diseases. So the expansion produces an altered form of the Huntingtin or HTT protein, which is referred to as mutant Huntingtin, which results in neuronal cell death in select areas of the brain and plaque accumulation. So Huntington's disease is a terminal illness. The symptoms include abnormal body movements, which is called chorea, which are jerky, random, uncontrollable, rapid movements, a lack of coordination. It also affects a number of mental abilities and some personality characteristics. So these physical symptoms commonly become noticeable in a person in their 40s but can occur at any age. If the onset is below 20 years of age, then it's known as juvenile Huntington's disease. It is a genetic disorder that does not have any cure, and symptoms are managed with medication and appropriate care. It's inherited as an autosomal dominant disorder, and you need only one affected allele from either parent to inherit the disease. So there's a 1 in 2 chance or 50% chance of inheriting the disorder from an affected parent. So however, the inheritance is a little bit more complex with these trinucleotide repeat disorders. In Huntington's disease, when the gene is more than 36 copies, the DNA replication process becomes unstable and the number of repeats can change in successive generations. So here's typical Huntington's disease pedigree showing an autosomal dominant pattern of inheritance. So the gene involved is the HD or Huntington disease gene. We already said located on the short arm of chromosome 4. And the end of the Huntington's disease gene has a sequence of 3 DNA bases, cytosine, adenine, guanine, or CAG, that's repeated multiple times. So CAG is the codon for the glutamine amino acid, which is why it's called the polyglutamine expansion. A sequence of fewer than 36 glutamine residues is normal. A sequence of 40 or more produces a mutated form, so that's considered abnormal. And the greater number of CAG repeats, the earlier the onset of symptoms. So here's a chart showing a normal individual has between 10 to 26 CAG repeats. Pre-mutation is 27 to 41, and a person that is affected has between 36 to 121 CAG repeats. So every child of a person with Huntington's disease has a 50% chance of inheriting the faulty copy of the gene and then getting the disease, because this is an autosomal dominant disorder. Pre-symptomatic testing is possible through a blood test, which counts the number of repeats in the gene. A negative blood test means that the individual doesn't carry the expanded copy of the gene, will never develop symptoms, and can't pass it on to their children. A positive blood test means the individual does carry the expanded copy of the gene, will develop the disease, and has a 50% chance of passing it on to their offspring. The full pathological diagnosis is established by neurological examination and through demonstration of cell loss, especially using a CT scan or an MRI scan. Can detect the Huntington gene CAG repeats through PCR. So in this case, this is a labeled PCR primers, and if you get a larger amplicon, that would indicate a higher number of repeats. So pre-symptomatic a positive blood test is not considered a diagnosis because it could be decades before the symptoms begin. Because of the ramifications on the life of an at-risk individual, there's no cure for the disease and no way of slowing it, several counseling sessions are required before an individual takes the blood test. A pre-symptomatic test is a life-changing event and a very personal decision. Currently, there is no treatment to stop the progression of the disease. However, symptoms can be reduced or alleviated through medication and specific care methods. So medication is used to alleviate symptoms. Nutrition is really important. People with Huntington's disease need more calories than a normal individual. There are studies being done on omega-3 fatty acids to slow down the progression of disease. There are other treatments that have been shown in some initial experiments to have promise, such as dopamine receptor blockers, exercise, antioxidant-containing food and nutrients, antidepressants, etc. The most hopeful future treatment is based on gene silencing. So since Huntington's disease is caused by expression of a single gene, silencing of the gene could possibly halt the progression of the disease. So there is a study done on a mouse model of Huntington's disease treated with a silencing RNA, and this achieved a 60% knockdown in the expression of the defective gene. So this study showed that the progression of the disease is halted. It's not completely stopped, but it is halted. Another trinucleotide repeat expansion disorder is idiopathic congenital central hypoventilation syndrome, or CCHS, which is a rare pediatric disorder characterized by inadequate breathing while asleep. This disorder commonly occurs in association with individuals who have Hirschsprung's disease, which is an intestinal disorder that causes autonomic nervous system dysregulation and dysfunction. A number of gene mutations have been observed, including a polyalanine expansion of the paired-like homeobox or FOX2B gene located on the short arm of chromosome 4. A triplet repeat expansion occurs in the FOX2B gene, resulting in multiple alanine residues. The severity of the disease increases with increasing numbers of repeats. Genomic imprinting is another disorder that is not inherited through your traditional Mendelian pattern. So in genomic imprinting, you get gene silencing due to the methylation of C residues as well as other modifications. So genomic imprinting occurs during the production of egg and sperm, and the phenotypic effects of imprinting are revealed in diseases in which the maternal or paternal allele is lost, which is known as uniparental disomy or uniparental deletion. Examples of genomic imprinting diseases are Prader-Willi syndrome, which is caused by a regional deletion or mutation in the paternally inherited chromosome 15, and the symptoms include mental retardation, short stature, obesity, and hypogonadism. Another genomic imprinting disorder is Angelman syndrome, which is a different disease phenotype caused by the same deletion or mutation in the maternally inherited chromosome 15. So symptoms include ataxia, seizures, and inappropriate laughter. So here is a methylation-specific PCR reaction to detect DNA methylation. So you can treat your DNA with bisulfite, which is going to convert your unmethylated C residues to U. You can then do a PCR reaction, and you are not going to get a product if you had methylation. if you get a product, you're not going to get a product if you have unmethylated C residues. So you have your U's there, you're not going to get a product. You will get a PCR product if you had your methylation of your C residues. Other methods can be used to detect DNA methylation, such as methylation-sensitive single nucleotide primer extension, PCR RFLP with methylation-sensitive restriction enzymes, and southern blotting with methylation-sensitive restriction enzymes. So genetic testing has limitations due to intergenic mutations in splice sites or regulatory regions that can be missed by analysis of gene coding regions. Also, therapeutic targets are phenotypic except for gene therapy. You can have non-symptomatic diagnosis where the disease phenotype is not yet expressed, and diagnosing these raises ethical concerns such as in Huntington's disease, where do you really want to know if you have the disease or not? You don't have any symptoms and there is no cure. Most disease and normal traits are multi-component systems. So it's not very often where you have a disorder caused by one single mutation in one single disease. So many diseases are multi-component, multi-genes, environmental factors, etc. So they're not as easy to detect using molecular or genetic tests. So that brings us to multifactorial inheritance and complex traits. So complex traits have no distinct inheritance pattern, and these include normal traits affected by multiple loci and or environmental factors. Quantitative traits are complex traits with phenotypes defined by threshold. So for example, in obesity, you have to have a BMI over 27 to be considered obese, or diabetes having a fasting glucose of over 126 micrograms. So your complex traits have thresholds. So you can do the same tests on them, but only if you get over a certain level would you be thought of to have that trait. So in summary, Mendelian, including autosomal recessive, autosomal dominant, and sex-linked, as well as non-Mendelian patterns of inheritance, are exhibited by pedigrees. Frequently occurring point mutations are easily detected through various molecular methods, including PCR, PCR RFLP, SSP, PCR, and southern blot. Non-Mendelian patterns of inheritance are exhibited by nucleotide repeat expansions, mitochondrial mutations, gonadal mosaicism, and genomic imprinting. Gene silencing on methylation of C residues affects the phenotype without changing the DNA sequence, and although molecular methods are ideal for detection of DNA lesions, molecular analysis may not always be the optimal strategy for laboratory testing.